Okay, so hello everyone. This is a continuation of our discussion for carbohydrate metabolism, and I think we are now going through the discussion for your glycogen. No? So glycogen in synthesis starts with the process we call glycogenesis. So, from its name, glycogen and genesis, no? So, meaning this is a creation or production of your glycogen. So, glycogen is a storage form of our carbohydrates in humans and animals. So, there are two ways that our carbohydrate is being stored in the form of glycogen, and this is a branch polymeric form of glucose, by the way. So these two types of glycogen in our body is muscle and liver tissue. So in muscle, it is a source of glucose for glycolysis, and in liver tissue, it is a source of glucose required to maintain the normal blood glucose level. So in glycogenesis, we have three steps, no? This is a metabolic pathway by which glycogen is synthesized from glucose 6 phosphate. So, the three process involves the formation of glucose 1 phosphate, so this one, and the formation of UDP glucose, glucose transfer to a glycogen chain, which is there's a branch transference, no, of our glucose. So through the process of different enzyme also. So this is the process of the whole process of glycogenesis. We have glucose through the process through the enzyme hexokinase or glucokinase in the liver. Remember, these are very specific. It can form glucose 6 phosphate, and through glucose 6 phosphate with the enzyme phosphoglucomutase, no, it forms glucose 1 phosphate, and through an enzyme we call UDP glucose pyrophosphatase, it forms UDP glucose, and with an enzyme glycogen synthase, it forms alpha 1,4 branch glucosyl units, and we have the branching enzymes so that it will form alpha 1,4 and alpha 1,6 glucosyl units. So, okay, so this is the step-by-step process by which it is formed, no, but I think it would be better, or it would be helpful if you draw a diagram such as this one for you to better understand the process of glycogenesis. Now we're moving to glycogenolysis. So from its name, glycogen and lysis. So meaning this is the breaking, no, sorry. This is the breaking down of your glycogen into glucose. So this is a metabolic pathway by which glucose 6 phosphate naman is produced from glycogen. So it's a bit a process that is reversible, no, or with the same diagram we can execute or show how this process are being formed. So there are two steps, which is step one, phosphorylation of a glucose unit, and step two, glucose 1 phosphate isomerization. So it would also be helpful if you would draw a diagram like this one, but I think the diagram that I've shown you works both ways for glycogenesis and glycogenolysis. So glycogen synthesis and degradation locally produce glucose 6-phosphate directly enters the glycolysis pathway. So low glucose levels stimulate glycogenolysis in the liver cell. So glucose 6-phosphate cannot cross cell membrane. Enzyme glucose 6-phosphatase found in the liver cell converts glucose 6-phosphate into free glucose. So this enzyme is not present in the muscle and brain cells. Okay, kasi sa muscle, ang ating enzyme doon ay si hexokinase, if you can remember the diagram that I've shown you a while ago. And free glucose is then transported to the brain and the muscle through the bloodstream. So this is a shortcut diagram for both glycogenesis and glycogenolysis. So as I've said a while ago, it can work both ways, itong diagram na pinapakita natin. So the path to glycogenesis starts with glucose, then glucose 6-phosphate, and then glucose 1 phosphate, and then UDP glucose, and then we have the glycogen creation. Okay, so this glycogen can be broken down, no, through the process we call glycogenolysis, in which glycogen will become glucose 1 phosphate, then glucose 1 phosphate will become glucose 6 phosphate, glucose 6 phosphate will become glucose. So remember that hexokinase is present in the muscle, while glucose 6 phosphate is present in the liver. Okay, so you may answer this or pause the video if you wanted to answer this multiple choice question. As a review, which of the following pathways is responsible for glycogen synthesis? Okay, so again, synthesis, genesis, production. So this can be a bit confusing if you're not familiar with your terms, but as you may know, glycogen, no, glycogen and genesis. So this one, the answer for this one is letter C. Okay. So next is neogenesis. Now we got a new term which is neo, which is new, no new or new form. Metabolic pathway by which glucose is synthesized from non-carbohydrate material. So it could be protein, lipids, no amino acids. So non-carbohydrate starting material for gluconeogenesis includes pyruvate. Now, of course, lactate from the muscles and from the red blood cells, as we have discussed from our previous video. We have glycerol, which I think this is from fats, no, and lipids from triacylglycerol hydrolysis. And of course, we have amino acid from our protein hydrolysis or from muscle protein during our starvation. So yes, no, pumapayat po or lumiliit, no, our muscle became atrophies whenever we are starved, no. So about 90% of gluconeogenesis takes place in the liver, and conditions in which gluconeogenesis occurs to replenish depleted liver glycogen stores, to convert lactate back to glucose, and to maintain glucose level when glycogen stores has been depleted. So the overall reaction is that 2 pyruvate plus 4 ATP plus 2 GTP plus 2 NADH plus 2 water will form glucose, 4 ADP, 2 GDP, and 6 phosphate and 2 NAD. So this is an overall reaction for your gluconeogenesis. Okay, so again, I've shown you this diagram a while ago, and side by side with my presentation, but this is a bit more complete process, no? So from glucose, ATP required in hexokinase. This is on your muscle part. It will become glucose 6 phosphate. Then we have another step, fructose 6 phosphate. Why? Because this is um, gluconeogenesis is the red one, while glycolysis is the blue one. No, so glycolysis happen using hexokinase and then glucose 6 phosphate. And then remember, the main product of your glycolysis is creation of ATP, right? Since it's glycolysis, the usage of glucose to create ATP or energy. So ang end product nito usually is pyruvate. No? In gluconeogenesis, is your pyruvate or non-carbohydrate source will form and become a glucose, which can be used by our body to create another energy. So this is gluconeogenesis. No? We also have this term called Cori cycle. You might encounter this on your exam. Gluconeogenesis uses lactate as a source of pyruvate. So lactate on this process, no, will have a lactate at the very bottom until it become pyruvate, so that the pyruvate will become glucose. So that's Cori cycle, no. Lactate is formed during strenuous exercise, and as I've told you on our part one of carbohydrate discussion, lactate is the main or lactic acid formations in or accumulation in the muscles, the main reason why we feel or why we have cramps, no. So lactate is formed during strenuous exercise. It diffuses from muscle cells into the bloodstream and is transported to the liver. So too much lactate is poisonous to our blood, no? And lactate is regulated pa din, of course. So enzyme lactate dehydrogenase converts lactate to pyruvate. So lactate dehydrogenase has many isoenzymes, and you will discuss this on your clinical chemistry, and you will notice that we have lactate or LDH 1 up to 5, and this can be an indicatory of your myocardial infarction if using our electrophoresis, no. We have a flip pattern on this enzyme, and this is very helpful, but not totally used as a diagnosis, no. This is just an enzyme that can be used with the other enzyme to give to monitor or to give a further diagnosis to the existing problem in the body. So pyruvate is then converted to glucose via gluconeogenesis, and the glucose enter the bloodstream and is transported to the muscle. So this is a gluconeogenesis nucleotide triphosphate change associated with two parts of the Cori cycle. Okay, so we have the glucose right here. Production using glycolysis. Again, glycolysis is transforming your glucose into a pyruvate, but the main purpose of this one is not to convert glucose to pyruvate, but to create an ATP out of the food source that you are getting. So, this is just kumbaga, byproduct, no? Itong pyruvate. But the main purpose of this process is to create an energy. Now, pyruvate will then be formed or converted into lactate, no, with the use of a reduced NAD, and then lactate in on the other hand, during our gluconeogenesis, um, will form or will be converted to pyruvate again, and this pyruvate, no, during gluconeogenesis will form glucose again, and then remember that this gluconeogenesis happen in the liver, and the formation of your lactate, as said, no, a while ago, lactate is formed through strenuous exercise. So it means it happened in the muscle, no? And the main communication channel for both of them is your bloodstream. So that is why too much accumulation of lactic acid in the body or in the blood is not healthy, because lactate, no, is a lactic acid. Other name for it is lactic acid. So there's a tendency that your blood will be more acidic due to accumulation of your lactate. Okay, so which of the following pathways is responsible for the synthesis of glucose from non-carbohydrate material? Okay, the answer for this one is gluconeogenesis. Okay, again, you can pause this video to answer this question. Where does gluconeogenesis occur in the body? Okay, we know the answer to that is the liver. Okay, so again, this is a summary of our terminologies discussed. No, glycogenesis is the creation of glycogen. Gluconeo, the creation of new or other form other than carbohydrate, other molecule to create glucose. Okay, and glycolysis is the formation of your glucose to make it pyruvate, and in return the main product being focused is our ATP, or the energy. Glycogenolysis is the lysis, no, or the breakdown, sorry, of your glycogen, no, into a glucose, so converted mainly, to be specific, glucose 6 phosphate, and glucose 6 phosphate will be converted using glucose 6 phosphatase into a glucose. So name ending with lysis means breakdown, and genesis means making or producing. Okay, so here are some totality of the major metabolic pathway that we have discussed. Okay, here are some practice exercises for you. You can pause this video if you ever wanted to review and read and answer this question. Okay. Okay, so another question. Okay. Okay, so let's move on to our pentose phosphate pathway. Okay, so this is a stress structure of NADPH. Okay, this PPP, okay, or is a pathway in which glucose 6 phosphate is used to produce NADPH, no? Ribose 5 phosphate and numerous other sugar phosphates. So NADPH is a reduced form of your NADP, or your nicotinamide adenine dinucleotide phosphate. Okay, NADP and NADPH are phosphorylated version of NAD and NADH, meaning it got an additional phosphate group on its main or in its base. So NADPH is essential for biosynthetic reaction pathway. So there are two stages, no, of our pentose phosphate pathway, which is oxidative and of course non-oxidative stage. Oxidative stage involves three steps through which glucose 6 phosphate is converted to ribulose 5 phosphate and carbon dioxide. Okay, non-oxidative stage has first step In the first step, ribulose 5-phosphate, which is a ketose, is isomerized to ribose phosphate, which is an aldose. Okay, so helps this. The purpose of PPP is to help meet cellular need. When ATP demand is high, the pathway continues to its end product, which enters glycolysis. Okay. Now, when NADPH demand is high, intermediates are recycled to glucose 6-phosphate. So the start of the pathway and further NADPH is produced. So it helps generate ribose 5-phosphate for nucleic acid and coenzyme production. So kapag sumosobra na yung glucose natin, yung ATP, no, of course our body has some way to recycle them and use them on other functions that is needed in the body, such as ito nga, see, nucleic acid formation and coenzyme production. Okay? Okay, so this is a problem again. We can pause this video to answer this question. Okay, so the answer for this one is a metabolic fat pathway by which glucose is used to synthesize ribose, 5-phosphate, and NADPH when needed. Okay, so this is a second, no, hormonal control of carbohydrate metabolism is a second major control method for controlling, sorry, carbohydrate metabolism beside enzyme inhibition by metabolism. So hormones that controls carbohydrate metabolism includes insulin, glucagon, and epinephrine. Of course, there's a lot more, no? If you can remember on your anatomy and physiology, insulin, no, is produced by your beta cell. My main mnemonic for this one is binsulin, no, of the pancreas, beta cells of the pancreas. And the purpose of insulin, as we know, is it decreases the glucose level in the body. While glucagon, my mnemonics for this one is aglucagon, which is released. This glucagon is released by our alpha cells in our pancreas. Now, and we have your epinephrine, which aside from being a hormone, is also a neurotransmitter. If you can remember, there's a lot more of enzymes available that increases the glucose level and in the body, but only insulin is the only enzyme, no, of course, that is involved, no, in a lot of contexts that decreases the level of glucose in our body. So one example is cortisol, no, there's a lot of enzymes available, so you will understand and you will learn more of that on your clinical chemistry 3. Okay, so this is a second major method for controlling carbohydrate metabolism aside from enzyme inhibition. Of course, you can always inhibit enzyme. That's a feedback mechanism of our body. So insulin is a 51 amino acid protein hormone produced by the beta cell of our pancreas, and it promotes the utilization of glucose by cells. So its function is to lower the blood glucose level, and release of insulin is triggered by high blood glucose level. Okay. On the other hand, okay, so the mechanism, hold on. The mechanism for action is it binds the protein receptor on the outer surface of the cells, which facilitates entry of glucose into the cells. So it produces an increase of rate of glycogen synthesis also. We also have our glucagon, which is a polypeptide hormone, a 29 amino acid, and it is produced by our alpha cells in our pancreas. Okay, and the principal function, of course, is to increase blood glucose concentration. Its mechanism is to speed up the conversion of glycogen to glucose in the liver. So it elicits the opposite effect of our insulin. Another one is epinephrine, which is produced in the adrenal gland. Okay, so these are aside from epinephrine, we also have norepinephrine on the same area, medulla area of your adrenal gland. And other name for epinephrine is adrenaline. So, or yeah, it is your adrenaline rush. Really, it is released by the adrenal gland in the response to anger, fear, or excitement. You know, you're familiar with the adrenaline term. So, this epinephrine, no, can be found at this adrenal gland, by the way, can be found at the top of your kidney. No, it's like a hat at the top of your kidney. So the main function of this epinephrine is similar to glucagon. It stimulates glycogenolysis. So it breaks down our glycogen in the liver, and its primary target, no, is the muscle Since this is a fight or flight hormone, it promotes energy generation for quick actions, and it also helps in lipid metabolism. Okay, so this is a stimulation of our glucose production by epinephrine. We have the epinephrine, the first messenger or neurotransmitter hormone attached to its receptor site. It will take effect with adenyl cyclase, and with the use of another ATP or energy, no, it will affect cyclic AMP, which is a second messenger. This is mostly the same pathway with other neurotransmitter, if you can remember. So this cyclic or this CAMP, our second messenger, will affect glycogen phosphorylase and will then help break down the glycogen to produce glucose 6 phosphate. Okay, so this is happening inside your cell. Okay, so this question, can pause again this video. What hormones controls carbohydrate metabolism? Okay, so the answer for that is letter C. Okay, so B vitamins and carbohydrate metabolism. As we know, we have niacin and fad, no, yung riboflavin natin, which we have discussed, no, we have, as well as the CoA or the pantothenic acid. We have discussed them on our previous discussion on our carbohydrate metabolism. And we know, I might have discussed also that they are B vitamins. So that's why B vitamins is very important in carbohydrate metabolism. No, that's why we need to intake a lot of B vitamins in our body, enable for us to have a better metabolism of carbohydrates. B vitamins are coenzymes in metabolic reactions, and B vitamins that are involved in carbohydrate metabolism is niacin, no, or yung NAD natin, and the reduced NAD. We have the riboflavin or the FAD, as well as the reduced FAD. We have the thiamine, we have the pantothenic acid or the CoA, we have the biotin, and of course we have the vitamin B6 in the form of your pyridoxal 5-phosphate. Okay, so here are how important these B vitamins are in the reactions of our carbohydrate metabolism. Okay, so this question, you may also pause this for this one. How many different B vitamins participate in the reaction involved in the carbohydrate metabolism? The answer to that is 6 B vitamins. Okay, so I think these are concept question. You may also pause this concept question if you wanted to read them. I'll just go over this one, so it would not take a lot of time from this video. Okay, so that's it, I think, for our chapter 24 or carbohydrate metabolism. I will upload this as part 2. Please don't forget to watch first the part 1 before you jump right in here. The part 1 is a bit longer than this one. Okay, so I hope you've learned something from this discussion. Thank you so much.